So we're going to segue very nicely into this session where we're going to talk about our local efforts to support translational science with some real-world examples as the panelists come down here. So you've already heard about what translational science is. We're talking about the scientific and operational challenges along the translational research pipeline and how we can make that pipeline more efficient, faster, more impactful. This is mandated by NCATS. Successful translational science projects provide insight beyond one investigator's work and facilitate translation of discovery into diagnostics and treatment across multiple applications and diseases. So it's a disease-agnostic approach to the processes by which we translate effective science into therapies. NCATS has put together a series of badges that represent aspects of translational science. And so when we have local efforts, we ask our investigators to address these particular principles in their projects. So as Bill mentioned, we've had an effort this year to provide supplemental funding to some of our investigators to come up with an aim that was translational science focused. So we took our investigators in our clinical and translational funding project program. These were all people who'd been awarded a CTRFP grant this calendar year, and we offered $25,000 additional funding if they could come up with an aspect of their project that they felt addressed one or more of these translational science principles, with a particular focus on breaking down barriers to efficient conduct of transformational translational research. Where are the hang-ups in this process that we all, as investigators here, encounter, and how can we locally and hopefully disseminated nationally address those barriers and reduce those barriers? So we have 3 panelists. We had 41 CTRFP recipients this year. We invited them all to apply for this supplemental funding. We received 21 applications and selected these 3 investigators up here as the recipients of our supplemental translational science funding this year. And so I'm going to ask each of these panelists to introduce themselves and talk about their particular translational science aim of their project. They'll give you a little background on the overall project first that's not so disease agnostic, and then talk about the translational science aim they've developed. So we'll just go down the row here. Maybe Mary, if you'd start.
I think I need a slide.
I've got your slide. Oh, is that your slide?
No, 2 more. Yeah. Thank you so much for inviting me and for the award. We have been focusing for the past few years on developing a health literacy informed visual key information page for informed consent. This actually came out of work that I was doing in my broader area of research when COVID happened and we all transitioned to virtual recruitment. And we were showing participants our informed consent documents on Zoom or trying to explain to them what we were doing in our research studies. And they were completely confused because we told them we were doing surveys or interviews, and it looked like we were selling their data. One person actually told us, "Are you selling our data to Google? I don't want to participate." And we went to our IRB, which I had tried for many years, and asked them, could we do this in a better way? And they suggested we use the key information section of informed consent because that was created with the Common Rule in 2018 that provides a concise and focused summary of your study and asks people to explain in plain language to participants the key information they would need to join a research trial. But most most investigators were just copying and pasting their entire consent document, shortening it to a few points and creating this longer text-based document. So we, with support from the ICTS, developed a visual key information page and support from Siteman Cancer Center, and our institution just wanted to implement. They said, well, great, you can use this, but we really wanted some data to show that it was actually working. So we tested it in an implementation study with rapid implementation and feasibility testing. We refined it with stakeholder and community partner engagement. We use real-world data to be testing it in 3 CTSAs in the country, and we're working on evaluating that now. With the translational science funding, we're actually going to be training people to do this themselves so that my team and I don't have to review every study visual design and create it for them. We want others to be able to use this in practice with support from our IRB. They don't want 100 different versions of a visual key information. They want people to use it in the way we've tested it and developed it. So this will launch in September. If you're interested, that's when our training will be held, so people can, I know some of you in the room are already using this, which is great, but that's the support for our translational focus.
Super. Lauren?
Hi, excuse me. My name is Laura McPherson. I'm in the program in physical therapy here. And my work is supported by both the KL2 program as well as the CTRFP pilot program. And we study how the nervous system controls movement in people with multiple sclerosis, which is a neurodegenerative and demyelinating disorder that can have profound effects on the movement system. And so we do this by looking at voluntary motor commands at the level of the spinal motor neuron. And this is after all of the upstream processing by the brain and the spinal cord where motor commands are most relevant to movement. So in the spinal cord, there's tens of thousands of neurons that converge on the motor neuron, but the motor neuron is the only output to the muscle to have the muscle contract. So all of the neural code that's required for movement is contained within motor neuron discharge patterns. It's kind of like Morse code, where it's a series of simple dots and dashes that has complex information. So we can measure motor unit discharge in humans non-invasively, and we can access that code for movement. So we can do that now through high-density surface EMG decomposition while people are contracting and get populations of motor unit discharge. So we take that information and look for different features within the discharge that correspond to fundamental components of the voluntary motor command, which is excitation, inhibition, and neuromodulation. So we apply this approach in our KL2 project to study neural mechanisms of motor heterogeneity in MS, which is a major barrier to developing mechanistically targeted rehabilitation interventions. And in our TRFP project, we began to explore whether this approach has potential as a biomarker of MS disease progression. So our approach is really a paradigm shift in how we study how the body controls movement. It's based on 40 years of basic science work, and in combination with enabling technologies, it's really accelerated its ability to translate the approach into humans. And it's really taking off in the field like wildfire. But a major logistical and scientific barrier is that we have no large database from people without neurological injury to use as a reference group. So as a result, every research group has to collect their own control data, which is usually a very small group that's not representative demographically, and it's not very robust as a representation of the non-injured state of the nervous system. So because we study MS, we have data from controls ranging from their 20s up until their 70s. And because we study heterogeneity, we have a very large sample. So we have like data from 80 participants rather than 10, which might be typical. So we started looking at this control data, and we found that there's a very big impact of age, which we kind of knew. But we also noticed that there's an age by sex interaction. So the effective age on our measures differs for males and females. And so for our translational science supplement, we will expand our control data set so that we have enough people from each decade that we can really make robust determinations about the effects of age and sex, so that the field can understand how to study and control for these factors. And then we'll share this data set with the research community so that everybody can use it and won't have to rely on regenerating a control group every time. And they can devote resources to collecting data from clinical populations, which, we never have enough data from clinical populations, and it will really help accelerate the efficiency and speed of the research if we can have this large resource. So I was really glad to have this funding opportunity because data sharing is always something that is in the back of my mind, like, we really should do this, but it's sort of like, who has the time, and there's a lot to do to actually make that happen, and so this has been a really nice catalyst to actually, initiate that process.
Super, thank you. And so last we have Christine.
Good morning. My name is Christine O'Brien. I'm an assistant professor in biomedical engineering. My lab tries to develop light-based tools specifically focused on maternal health applications. And for my original ICTS CTRFP project, we were trying to develop a tool that could possibly identify preterm birth risk by specifically studying cervical mucus. And so in the picture here, you can kind of see a schematic of a high-quality, non-permeable mucus barrier that prevents ascending infection through the cervical canal to the uterus. Versus a highly permeable, likely to be very hydrated, very low viscosity mucus sample that is permeable and could lead to preterm birth. So about 40% of spontaneous preterm births are associated with infection and likely ascending infection. So this is a huge unmet need. And there was some really interesting work by colleagues in Boston that were able to show the mucus, and that's this figure on the bottom left, that the mucus viscoelasticity specifically was very, very different in patients that were at low risk versus high risk for preterm birth. And the way that they did this, for this particular test, they needed a very large sample of mucus, but then they also did tests where they used a really expensive, complicated rheometer instrument, which, for example, at WashU, we have one of these. It is in a special materials science building operated by technically trained staff. It takes weeks to get trained on how to use it. My students have gotten trained. It's arduous. And so it's just not a very clinically translational measurement for something that could be incredibly useful clinically. And so we were trying to think, we do light-based diagnostics. Is there a way that we could just shine light on the mucus sample and get that similar information? And so that was the goal of our original CTRFP, was to essentially extract mechanical information from the mucus just by shining light on it. And so you can see the kind of the schematic of what we envision that system might look like one day. And then for the translational sciences supplement, we tried to expand out to how do we make this more generalizable beyond just preterm birth. So mucus is going to be covering all sorts of membranes, so things in the lungs, the GI tract, and essentially the permeability of mucus in these different organ systems is related to a host of different conditions, such as inflammatory bowel disease, cystic fibrosis, et cetera. And so we were wanting to see, can we push this a little bit further, make it more generalizable, and also see if we can quantify hydration of the mucus, which is particularly linked to mucus permeability. And so that is, I guess, the extra piece that we have proposed in our translational sciences supplement. So linking our mechanical properties, our hydration, to the actual mucus permeability, which will be hopefully generalizable to a host of different disease states and I guess, clinical applications.
Super, thank you. That's great. So I think I'm gonna ask a couple of questions of our panelists, but if you have questions, please raise your hand and we can incorporate your questions as well. Sitting here listening to these explanations, it strikes me that all three of you are dealing with scalability in these supplements. Sort of, how can you take the research you're doing locally and make it applicable to a broader group and more generalizable? So can you talk about the challenges you anticipate as you try to scale your projects? We'll start with maybe Christine and work backward.
Sure, thank you for that question. So as an engineer, one of the first things we think about is cost and size. So right now we do have a working prototype, but it's about this big. And we're hoping to have something that, I don't know, looks more like the size of like a small toaster, for example. That would be great. It could just be sitting in a clinic. We also are trying to think about simplicity. So we want there to be very few steps. So we could ideally, I think our goal is that we could publish, like a Jove paper on like, how could you build this yourself and have it be as simple as possible, show all of the components that we would need. So try to have as simple components as possible, easily accessible off-the-shelf components that could be assembled. We could include like 3D printing files that could be used to like assemble it together. And include data analysis code so that people could enter in their data, they could get the mechanical properties out, they could get the hydration levels out, et cetera. So those are, I guess, the things that we are thinking about. I haven't thought much about scaling beyond the research, but I also think if we really do want to continue thinking about scaling like beyond just to researchers, we would look at things like SBIR funding, trying to see is there commercialization potential, et cetera. So those are I guess, the thoughts that I'm thinking about right now.
Great, Lauren.
So for me, if I'm going to be successful with this project, I need to have people in the field adopt my data, which I think, at least speaking for myself, is a little hard to use data that other people have collected, because it's like, well, I want to make sure it was right, and I know it's right if I do it myself, So one thing is building the trust of the people in my field that we did this well, and fortunately, I have good relationships with people in the field, and I think I have that, a good reputation for quality, but also thinking then in my lab, trying to anticipate things ahead of time in terms of our inclusion-exclusion criteria, in terms of what things that we measure that could potentially be confounders, so that we can kind of head off issues ahead of time. So if we discover that, if somebody has had a knee replacement, then that might affect our measures in this way. So do we exclude them now, or do we just write down that they had it? And so just trying to anticipate ahead of time anything that might affect the applicability and accuracy of our data, so that it really serves the purpose of being something that people do not have to replicate.
Great. Mary, additions to that?
Yeah, for us, our biggest challenge with scalability has been identifying the core components of the intervention and identifying the modifiable components. Because we're doing this at multiple CTSA sites with different IRB regulations and different investigators, there are slight modifications that have to be made to meet local culture and regulatory requirements. But there are certain things that we did not want to change because we did extensive formative work to find out what works best for whom and under what conditions. So we kind of worked to train people on those core components so that each IRB reviewer wasn't reviewing 10 different versions of something and then making it much harder for them to find the details that are necessary for the study. And then the things that are modifiable are slight formatting adjustments or versions of the information, video training, things like that. And so far that's been functioning well, but we've done a lot of engagement with the regulatory community as well as the investigators and community partners. So beyond the sites we've used, we know there may be some uphill challenges with scaling out.
Great. I'm watching for questions in the audience. So, you're talking about impacting the process by which people can further their science using some of the technologies and techniques you're developing. How do you measure success? Do you have a testable hypothesis, or are you looking more at an end result upon project completion? What does success look like in a translational science endeavor? Laura, maybe we'll start with you.
Sure. So certainly in terms of the open data set, I'm planning to disseminate it in such a way that I can keep track who's using it, encourage them to cite the data set. I know that there's different avenues where different journals that will publish open data sets, and so that's a way to track citations of it. And then I also hope to communicate with researchers as they're using it. So for example, I have some collaborators who are submitting a P award for looking at aging and spinal cord injury. And some of the initial feedback they got from reviewers is that they wanted more control data of people who had been aging. And they're like, well, we don't have time to collect a lot. And I said, well, I have this enormous sample. I don't have a database yet, but you can use these graphs, in the grant. And so keeping track of those types of dissemination. So I hope with the open data set that will be somewhat quantifiable.
Christine, you want to?
So I think the first thing that we typically do to try to measure success is just do a bunch of validation studies to make sure our device is working as expected. So for example, we make synthetic mucus right now, and we can run all sorts of tests on it. We can kind of fully characterize it. We can measure its permeability. We can exactly control its hydration. We can exactly control its mechanical properties. So that helps give us confidence it's working as expected. I think when we get more to the translational space where we are measuring human samples and mucus is a little bit more hard to come by, we are going to have to start looking at outcomes and ideally also enrolling healthy control patients and again looking at outcome measures. I also think we could probably start trying to recruit a number of other disease states where perhaps mucus is more prevalent, where we can actually collect enough mucus to still run those validation studies. In some patients, we are able to gather quite a bit of mucus. So when that is possible, still running those validation studies to make sure we are getting what we expect to see. But yeah, I think that the farther scale, we do have to start looking at outcomes.
For our study, we have multiple levels of outcome measures we're using. We're using the translational science benefits model to look at implementation outcomes. We're also using a measure of reach and also reach by study type, because there were lots of questions about can this work for all different types of studies. We're also looking at time to review studies, because that was a big barrier that investigators were concerned with. Will it take extra time for my study to get approved that I don't have? So we actually worked with in the formative work to ask people what was important to them to make sure we were including it in addition to our standard investigator decided measures based on our frameworks.
That's great. So I hope everyone in this room knows, but if you don't, the Translational Sciences Benefits Model is a WashU developed tool to translate your work into a more global perspective on your impact in multiple domains. And I encourage you to go to that website if you haven't explored it and how it might apply to your work. We have some questions over here. Ruth?
Thank you so much. To ask you, did you have sites that you approached to participate that you weren't able to get to the table for participation?
Do you mean particular studies that we couldn't get
to
participate?
Exactly.
No, so far every study that has that we've approached or they've approached us, we've been able to get approved, from basic science in cancer through memory and aging at the Alzheimer's Center. We have strategies to suggest ways to summarize findings and the procedures.
That's very, very impressive.
Yeah,
Very
yeah, it's
impressive.
working well, and it took a long time to get the IRB comfortable with where we submit it. It always gets submitted as part of consent, not as a supplemental document, because it's being used as consent. So it's very similar to how people were already summarizing in text. It's just that it's an easier way for participants. And then the studies that have started became our champions for other studies, because their participants were saying, I'm pasting this on my refrigerator so I know what to expect on my follow-up. And they had taking it out of their wallet, all folded up, to remind themselves at their next visit what they were doing. We were really fortunate that our cancer center invested in it originally as well, because they had studies that they were looking at, like phase 2 treatment trials, that they said, hey, try this one, see if it works. So we could get that data.
No, that's terrific. Laura, can you describe just very briefly what's involved in the assessment?
Sure. So we have a standardized assessment where we have somebody sit in a testing chair And they have so we test the ankle muscles, just as an example. So they have their foot sort of connected to this machine that will measure the joint forces that are generated as they dorsiflex and plantar flex. We have stickers that go on the muscles that are high-density electrode arrays. So there's 64 channels that measure EMG activity. And so we measure that while they do different tasks, and then the processing is offline. And then that is somewhat time-consuming in terms of signal processing and cleaning the data. There's the decomposition into action potential spike trains is semi-automatic, so there's an automatic stage, and then you have to go in and manually kind of correct a lot of things. So the reason why, in my field, it's like you want to make sure the data is right, because you really have to know what you're doing to get something that you can trust, because you can get results, that may or may not be correct. And so fortunately, when I was a trainee, I was able to start using this technology right when it was coming into human use. So I was able to train with people who were like, okay, here's how you have to do it very rigorously. And so that's been a big benefit, to still be connected with those people so that I trust what we're doing. And then, trying to train my lab staff, and then we're about to have a big turnover, so, training people as we go through. And so I think now that I'm explaining this, part of the dissemination of the open data set will probably be a lot of education about how to analyze their data so that it is appropriately comparable, to our data set.
Yeah, I can see a challenge here, and it totally sort of underscores the point you made earlier about the challenge of just assuming anyone can do this, and why you want to look at the data. Yeah. And much as we started with neuroimaging and sleep data many years before. So one of the barriers for you is how then can this be implemented in other labs easily, or they would obviously have to have similar expertise. And is there a future in which you would see this being able to be all more automated?
Potentially, yeah. So and I should say, like, this approach was not just one I developed, but there's sort of a team of us in the field. And so there are people working on the algorithm development itself to try to improve that. And there are open software programs to be able to do the decomposition now. And so some of them are working on the training for that part of the analysis. And so I think coordinating with all of them, and having their toolbox linked to my data set and my data set, and making sure that all of our tips and tricks and, are sort of synchronized, will be something that's valuable. And different groups host, like an annual tutorial of come to this university and have a boot camp in how to do this. And so I think, coordinating all of our efforts will be very important. Because, like I said, people are using this now, and it's like going like wildfire throughout the field, so that it's a matter of pulling back a little bit and making sure, the speed at which it's been adopted sort of is a little nerve-wracking to some of us who are really, in the details, because we're like, hang on, let's make sure everybody's doing this in a way that the data will be accurate and valuable, and not just add noise to the literature.
Very helpful, because there's a huge interest in motor components for many disorders in psychiatry now in a way we didn't before, and ways to assess that and address that are going to become, I think, just more important.
Yeah, thank you.
Thank you, Ruth. We've got a question behind you. Tim has a question. We're moving the microphone. I'm hearing themes of rigor and reproducibility from all of you, that to effectively scale and promote more rapid translation, there has to be supreme attention to rigor and reproducibility. But I'm also hearing a really important theme, which is that your sort of universe of collaborators for all of you has grown almost exponentially with this focus on translational science. So I hope that everyone is seeing the potential of this sort of shift in mindset. You can't lose your disease-specific research focus, but to incorporate also this attention to barriers to translation and how you and your research program might tackle some of those and the potential that has to really expand your scientific arena. Tim.
Yeah, thanks. And this is really exciting to hear about these great projects. And I want to invite, following on the question from my sister-in-law, Mary, if you could, your slide when you put it up there on health literacy, I couldn't read the fine details, and I want to invite you to talk a little bit more about the health literacy aspect of it, because the work that we did years ago on the ACA, when we looked at health literacy, if I recall the findings correctly, it wasn't the health literacy component that mattered so much, but it was the math literacy component. So, if whatever direction you want to take this question, if you want to talk about how you're going to incorporate health literacy in your work.
Sure. Thank you, Tim, for the question. We have been requiring a readability level of all of our tools. We also have health literacy principles in terms of font size and display. We give people standardized icons that we've tested with our community partners. We also just did a side project when I presented at a health literacy area research conference. Someone asked, do you need these icons? Why include them? Why not just include the text? And we thought, well, I don't know. We thought people like pictures, so we can test it and see. So we did a side project with an online panel and randomized people to the icon version with the boxes without the icons, and then just plain language text, all the same text, just randomized to different groups. And we found that there was no difference in their knowledge, their engagement with the materials, any of our patient outcomes except preference. They liked the icons. They thought they looked better. But it was about 70% of people. 15% of people said, it's distracting and it doesn't look professional to me. And it was older adults that said, I'm used to seeing this in text. And now when you put those pictures, it maybe looks a little less serious. So there were a minority of people, but it was more likely in older adults. So we're still using them, but we're thinking about how to measure that. So those are some of the health literacy considerations.
Bill, last question.
Well, last three questions are comments. I mean, one of the things Mary actually has progressed this to involve other CTSAs, and as I was listening to all of you, I was thinking, one of the things we ultimately hope to be able to do is take this sort of work and scale it not only at this institution, but ultimately to be adopted by the clinical translational research community in general. And so, Laura, one of the things I was thinking about was, and I wonder, have you thought about the fact that, when we funded this, we were very intrigued by the notion that we would develop a control group that other investigators could use. Now, initially that would be here at Washington University. But if this was to be reproduced by several CTSAs, so the ultimate outcome and the paper and the main paper that says this is normal, like a Tanner scale for motor, came from 4 or 5 CTSAs. Do you think that would help the field in some way of saying, we don't now have to reproduce the wheel every time we do a small study?
So if it came from different CTSAs because the numbers would be bigger or because it would be more investigators endorsing it?
It's a combination of, you get the numbers, you get the reproducibility because you're not relying on one lab.
So you're saying every CTSA or every group?
Well, we can't get all 60 to do it.
No, I mean, the participating ones, you're saying it would be reproduced at every university that was involved.
And you also then have a community that is advocating not just a single university community, but...
that would absolutely help. I think the problem is funding. So...
Well, CTSAs can be, as a network, can be creative.
Yes, yeah, well, yes. So I guess to convince my collaborators to do this, because the numbers that we're collecting here are enormous relative to the field. We have like 65 patients with MS, 80 controls, and typically studies are like 20 and 20.
I'm not disputing that they're enormous for the field.
Yes,
But they're not enormous.
No. So, but I mean, when I go to if I went to my collaborators and said, hey, would you collect 80 controls too? And then we can all, they would say, well, that's a lot. How am I going to have the money and resources? But if that were addressed, if they could go to their CTSAs and similarly get work, then yeah, it would be amazing to be able to have, more senior investigators, people who have repeated this at multiple universities, because in the data that we've seen so far that has been collected at universities, it is very repeatable, at least in small numbers. So yeah, I think that would be great to have it be more of a consensus from the field rather than just, me saying like, here, this is good.
And I would just say, maybe not having them all create their additions to your data set, but use your data set and say, it worked great, right?
Yeah.
And then it lends sort of this heft to your claim that this is a data set that can be used as a comparator, outside of your lab.
Yeah, these are all really helpful thoughts that I'm sure I will ping you as I go on this so that I can keep getting.
Please do.
Yeah.
Just to Bill's point, there are CTSA studies. The CTSA led out of the Medical College of Wisconsin by Risa Shaker has a study ongoing, Christina, on bringing in women who are pregnant for their appointments and getting some indicators, because they're in rural environments, of when they might need to come in sooner. Even simple things like, blood pressure, and how do you put a system in place for whom this might actually be very interesting for their local physician to have access to. And just, it's more, I think, what Bill is alluding to is thinking, are there other entities across, other studies ongoing across CTSAs that might be particularly interested in what you're doing and hearing about it, and maybe then forging some collaborations in the future?
And I know Nancy wants to close the session because she probably like me.
No, I really don't.
No, but I, just for Christina, I actually wanted to get back to a point that Nancy raised. I mean, one of the things that intrigued us when we were reviewing the proposals was the idea that you would take the work you were doing, which is very novel, and ultimately, hopefully, will be something that can be useful in clinics, beyond cervical mucus to mucus in general. But what my question is, that was the intriguing part to us. Has that opened new avenues for you as an investigator? Have you started to talk to the gastroenterologists and the pulmonologists and so on, that to start developing these collaborations?
Yes, that's a great question. I would say I have forged relationships with mucus experts and hydrogel experts. That's kind of where I've started. I have a very big team of maternal fetal medicine specialists. We have partners at Tufts University. We've got partners here. But specifically speaking with the GI and the pulmonary, we haven't begun those conversations yet, but it's hopefully in this month we will be starting those conversations.
So for you and for other investigators sitting down here, that's a role that the CTSA can help with.
Oh, perfect.
That is this sort of, for want of a better word, dating. We can convene these sort of, that you're doing very interesting, because one of the challenges I think at big universities, and I'm sure it's the same, I know it's the same elsewhere, is that people don't know what's going on in their own place.
Yeah, very true.
And so, we do have, we have expertise in cystic fibrosis. We're one of the major cystic fibrosis centers in the country, as an example. I am pretty sure that there are investigators there, we talked about whether you could better phenotype patients and determine, and I'm just taking it out of my head, who might need earlier consideration for transplantation by looking at their mucus. That would be an intriguing area of investigation. So come to us And if you have, you have an idea and you think it's scalable, or you think it could be brought to other investigators, and we can help with that.
Thank you.
So I'll just close by saying that to those pulmonologists and GI doctors in the audience, come and talk to Christine at the break. But also, I hope that this session has stimulated in you some thoughts about how you might add translational science to your research program, and also think about how the ICTS might help in that endeavor. We are eager to hear how we can help spur this type of work more on campus. And talk to me, talk to Bill, talk to anybody associated with the ICTS about your ideas about how the ICTS in particular can get involved in facilitating this type of work, because the NIH is super interested in this. And we have a renewal of our grant due next year, so we're really looking for the innovation and the ideas around translational science on our campus. So I'll stop there. We have a 10-minute break. Thank you all very much. Maybe 5-minute break. So thank you.